mechanisms which transport substances through the cell membrane. Now, the first thing that you need to understand, for you to even understand how transport happens, is the composition of the cell membrane. The cell membrane is made up of a lipid bilayer. So these are actually phospholipids which I'm drawing. The head is polar, it has got a phosphate, and then the tails are non-polar, they contain fatty acids. So you have got one part that is facing the outside of the cell, that's extracellular fluid. The other part of the heads are facing inside the cells, where you have got the intracellular fluid, ICF. Now, the cell membrane, because of these tails, is largely non-polar. Okay. Therefore, substances that are non-polar can pass through the cell membrane without a requirement for energy. Because remember, like dissolves like. So non-polar substances can pass through the cell membrane. And that takes you to the different types of transport that happens in the cell. There are three main types of transport that happen in a cell: passive transport, also known as diffusion, active transport, and then there is vesicular transport. The passive transport or diffusion basically happens through the lipid bilayer because it is non-polar. So substances that are also non-polar can just pass through the cell membrane without the requirement for energy. So that's basically passive transport. You don't require energy. So passing from a region of high concentration to low concentration down a concentration gradient without the requirement of energy. We have got, so passive diffusion is divided into simple diffusion, which is basically the movement of small nonpolar molecules directly through the lipid bilayer itself. Okay, that's simple diffusion. For example, these are small molecules because the cell membrane contains only small spaces where the substance can pass through, and they are non-polar because polar ones can't pass through the cell membrane since it's non-polar. Examples we can give are gases like oxygen and carbon dioxide. Facilitated diffusion is going to be for larger molecules which are polar. So for these ones, they can't pass directly through the cell membrane. So that's why they it is facilitated. So what happens that in the cell membrane, you have got there are two types of proteins. There are those proteins that run from one end of the cell to the other end of the cell. Okay, this is the cell membrane which I've drawn. This is a protein, and this protein that run from one end of the cell to the other end of the cell, we call it a transmembrane protein. Okay, the transmembrane protein, because it passes through the membrane. And what happens that this protein is going to now allow those substances that are charged, which can pass through the plasma membrane, and those which are larger, which can pass through the plasma membrane. So that is facilitated diffusion. So there is actually a protein or a carrier or a channel through which substances will pass. And then, of course, you have got osmosis. Now, osmosis is specifically for water, where water will be moving from high concentration to low concentration down the gradient. And the thing is that for water, I've said down the gradient, that is a region from a region of high water potential to low water potential. In other words, from a region where water is a lot, okay, to where water is little. Now, where water is a lot, this region is actually having low concentration because water actually dilutes solutes. Okay, so it's from a region of low concentration to a region of high concentration. But now simple diffusion is movement from a region of high concentration to a region of low concentration. That definition is dependent on what is actually moving. If it is the water that is moving, it is from a region where water is high to where water is low. But because water dilutes things, where water is going to be a lot, there's going to be a low concentration because concentration is basically found by the number of solutes over the volume of solution, which is in this case water. Okay, so water uses also pores which we call aquaporins in the cell membrane. For other substances, so that's what happens. Facilitated diffusion requires a channel, but what differentiates it from active transport is that there is no energy. Okay, even if it requires a channel. Active transport, on the other hand, is where substances will be moving from a region of low concentration where they are few to where they are high. And this is because you are going to be using energy to go up a concentration gradient. Okay, so it requires ATP, that's energy. And you divide active transport into primary active transport and secondary active transport. Primary active transport is where you are using ATP direct. For example, let's say you want to transport sodium, okay, from one end of the cell to the other end of the cell. For you to transport this sodium, you require energy directly. So what happens is that the channel through that will be transporting sodium is going to break down ATP and then you transport sodium. So there is need for for energy directly. An example that we can give is that, so we can give three examples. We have got the sodium potassium pump, which is found in all the cells. You have got the calcium transporter. You have got also what we call the proton pump. This proton pump is present in the kidney, also in the stomach. It's actually involved in the production of hydrochloric acid. So secondary transport uses the energy stored in an ion gradient created by a primary active transport. So for example, you want to transport, let's say glucose. Okay. What happens in this case is that you are going to be transporting sodium using energy. So the transport of sodium is going to be requiring energy. So ATP is going to be broken. And then glucose is going to take advantage of that ATP that sodium is using. Take for example, you want to move from school to town. You book a yango and you're only alone. I'm your friend. So what I can do is I can just ask you, since you've already booked a yango and I'm also heading to town, can I just jump on it also? For me, I'm not going to pay because you've already paid for it. Okay. So in that case, I'm the glucose. I'm not requiring any energy. And then you in that case, you are the sodium, you are requiring energy. You are paying. Okay, so that's what we call secondary active transport. To transport another molecule using the energy of another molecule that you are transporting. Okay, so that's what happens. Examples that we can give is the sodium glucose co-transporter. This is in the proximal tubules of the kidney. Okay, proximal convoluted tubules. You have got the sodium amino acid transporter also in the proximal convoluted tubule and also the small intestines. There's a sodium potassium 2 chloride co-transporter, and this happens in the ascending limb or the loop of Henle. Okay. Now, these three guys are going to be transported in the same way. In other words, the same direction where sodium is going to be going. That's where glucose, the amino acid, the potassium, and the 2 chloride ions also be going. So we call that as a co-transport, also known as a symport. Otherwise, let's say if you are moving sodium from this direction in this direction, and then there's another molecule this side which will be moving in this direction, but you are using the energy of sodium. In that case, we call that as a counter transport or an antipode. It's still a secondary active transport, and the examples that do work by that mechanism is calcium sodium exchange in the muscles, and then the sodium hydrogen exchange, okay, in the intercalated cells. When you come to the kidney again, the collecting ducts, and the distal portions of the distal convoluted tubules, they contain these cells, the intercalated cells, that will be able to secrete the hydrogen. So these are examples. Okay, that's active transport. And then vesicular transport, this involves the transport of large molecules or particles using vesicles. So what happens is that you have got a cell membrane like this. Okay, you have a cell membrane like this. Let's say a bacteria comes here. What happens that the cell membrane is going to go in like that, and then the bacteria is going to be eaten inside, and then it will close up. And then what you see is that now it will break off from the cell membrane, and then the bacteria is inside. We call this thing that has formed as an endosome. This endosome can fuse with a lysosome to form what we call a phagolysosome, and then the bacteria is going to be broken down. One like this, which I've described, we call this as phagocytosis or cell eating. Okay. And then there's also what we call cell drinking. For cell drinking is pinocytosis. What will happen is this: the cell membrane, instead of it going down like that, it will go like this. You've seen that it has gone outside, and then the bacteria is going to be there. It will be eaten, and then you still also form a phagosome. There's also what we call the receptor mediated endocytosis or clathrin mediated endocytosis. And what happens for this one is basically you are going to have, this is the cell membrane, okay, which will be like this, and then you have got receptors here, which we call uh, clarithins, and these receptors are going to be the ones that will attach the organism that has to be eaten. So the organism has attached there, or a microorganism has attached there, attached there, and then at the end, this top is going to close, and then this phagosome will form, and then it will go and join with a lysosome. All right, so that is what happens. Does that make sense? Is that okay? All right. So question 2 is saying, using a suitable example, explain clearly negative feedback mechanism. Well, negative feedback is just a regulatory process. Okay? It's a regulatory process that tries to make sure that homeostasis is maintained. Let's say, for example, we have got the hypothalamus, and then the hypothalamus releases hormones that control the pituitary gland. The pituitary gland now also releases hormones that will control many other glands. In this case, let me use the thyroid gland. Okay. Now, the hypothalamus releases what we call releasing hormones. So there is the thyroid releasing hormone. This will stimulate the anterior pituitary gland to release what we call the thyroid stimulating hormone. And then what happens now? The thyroid gland is going to now release the thyroid hormone. The thyroid hormone is made up of T3 and T4. Now, as the levels of T3 and T4 gets to increase, you can see that they are increasing. What this does is that these guys are now going to go back and inhibit the thyroid stimulating hormone and the thyroid releasing hormone. Okay, they are going to go and inhibit them, because as long as these guys are still being produced, you are going to be having more and more and more the thyroid hormone, and that can lead to hyperthyroidism. So you want to maintain homeostasis, a constant internal environment. So that is what is going to happen in that case. So once TSH and TRIH is inhibited, it means that there is no information to stimulate the thyroid to release more the thyroid hormone. And so a constant internal environment is going to be maintained in that case. So in this mechanism, the negative feedback mechanism, you can see that an increase leads to an inhibition. And what happens is that if there will be a decrease in the thyroid hormone, that will now lift off this inhibition. Okay, this inhibition is going to be out. And so it means that the thyroid stimulating hormone and the thyroid releasing hormone will now be high again. And that will come and stimulate the thyroid gland to release more of the thyroid hormone. And that still maintains homeostasis. Okay, it's different. This is negative feedback. It's different from positive feedback. What happens for positive feedback is that, let's say what happens in oxytocin. Okay, the posterior pituitary gland is going to release what we call oxytocin. This oxytocin is going to go on the uterus. The uterus will start to contract. Okay, so there is a stimulation on the uterus. It's contracting. That contraction on the uterus is going to go and stimulate the anterior pituitary gland to release more of oxytocin. Instead of inhibiting the release, it will stimulate it to release more of it. That is because the uterus needs to continue contracting so that the baby can be out. Okay, imagine the uterus just contracts a bit and then it stops contracting and then the baby is about to come out. That is going to be a problem. The baby can die. Okay, so this is what positive feedback does. The hormone is released, and then the release of the hormone stimulates even more of the hormone to be released. That's positive feedback. Negative feedback is the release of the hormone is going to inhibit the release of more of the hormone. Okay, so that's what happens. So in this negative feedback loop, we have got a stimulus. In this case, it can be low thyroid hormone, depending with what hormone you are discussing. Okay, that will be the stimulus. And then there's going to be the response by the hypothalamus. So the hypothalamus is going to, so let's say if there is a low thyroid hormone, the hypothalamus is going to respond by releasing the thyroid releasing hormone. And then the pituitary gland will also come in and release the thyroid stimulating hormone because of the instruction from the thyroid releasing hormone. And that will now, the thyroid stimulating hormone is going to stimulate the thyroid gland to release T3 and T4. Once the T3 and T4 are high, those are now going to go and inhibit TRH and TSH. That will be the negative feedback. Okay, so that is what happens. Does that make sense? Is that okay? All right. So the next question, number 3, it's saying what is meant by shift of oxygen dissociation curve to the right? Enumerate the causes of such a shift. Is this shift advantageous? So there are main questions there. Now, when you look at the way hemoglobin is, because we're not talking about the shift of oxygen dissociation curve, we are basically talking about hemoglobin. So hemoglobin is made up of two things. There's what we call heme, and then there is a globin. Okay. The heme is made up of two things. Globin is actually a protein. Heme is made up of iron, mostly as iron 2 plus. If it is iron 3 plus, we call that as methemoglobin, and it doesn't even have the potential to carry oxygen. Okay, so there is iron, and then there's what we call protoporphyrinogen. Protoporphyrinogen 9. So what happens that this ion is actually on the middle, and then you have got these rings like that. There are 4 rings, which are the protoporphyrin rings. But what is important that I want you to understand is that this ion is what has got the ability to bind the oxygen. Okay? Ion is able to bind oxygen, and it will be able to bind 4 molecules of oxygen. So that's what happens. Right? It can be able to bind 4 molecules of oxygen. Now, as long as hemoglobin, it's actually the ion in hemoglobin that binds to oxygen. But forget about that now, since we already know. So I'm just going to be using HB. So when the hemoglobin is bound to one oxygen, remember it's 4 of them that binds to hemoglobin. So 1 out of 4, that is 25. So this is 25% saturated. If the hemoglobin is bound to 2 oxygens, that is going to be 50% saturated. If it is bound to 3 oxygens, that is 75% saturated. And if it's bound to 4 oxygens, that is 100% saturated. Okay? Are we getting the sense? All right. Okay, so that is what happens. Now, look at this. What is happening is that when hemoglobin, you want hemoglobin to bind to oxygen if you are in the lungs. Okay, let's say this is the alveoli, and then so that's the alveoli, and then this is a blood vessel. Okay, this blood vessel contains HB. The oxygen is actually coming from the air. So when it comes like that, it should be able to bind to HB. This is what you want when you are in the lungs. Okay, you want the oxygen to bind to the hemoglobin. But once this hemoglobin has bound to the oxygen and then it has gone and then you reach to the tissue, this is a blood vessel and then this is the tissue. You want the oxygen to enter the tissue. So what should you do? You want the hemoglobin to let go of the oxygen so that the oxygen can diffuse into the tissue. Okay, so this is the basic thing that happens. Now, there are some things that will promote the oxygen to get away from the hemoglobin so that it can go and enter into the tissue. And such things include number 1, remember the tissues were actually working, they are breaking down, there is metabolism happening, so there is a lot of hydrogens that are being produced. Besides that, remember tissues, there is respiration that is happening and such. So a lot of carbon dioxide is being produced. So the partial pressure of carbon dioxide. Besides that, you also have what we call 2,3 bisphosphoglycerate. Bisphosphoglycerate. Besides this, also high levels, elevated temperature. Okay? So if you have got high levels of hydrogen, high levels of the partial pressure of carbon dioxide, High levels of 2,3 bisphosphoglycerate, temperature. Now, if you have got a high level of hydrogen, hydrogen is actually going to cause pH to be acidic. This means that you have got a low pH. And that's actually the same thing that happens with partial pressure of carbon dioxide, because what happens is that carbon dioxide can react with water, okay? In the presence of the enzyme known as carbonic anhydrase, you are going to form carbonic acid. This carbonic acid can further break down into hydrogen and bicarbonate. So you can see that you are still forming hydrogen. In other words, if you have got a lot of carbon dioxide, just means that you have got a lot of hydrogen. Okay. So these factors that you can see here, they promote the removal of oxygen from hemoglobin because they also compete for the same position. So when these guys bind to hemoglobin, it means that oxygen will not be able to bind there. So in other words, the binding of these guys to hemoglobin reduces the affinity of hemoglobin to oxygen. And so oxygen can now go and bind to the tissues. It can be released and go to the tissues. And that's what happens. So for the hemoglobin saturation, okay, the oxygen saturation curve, this is how it looks. The y-axis represents the saturation, how much oxygen is bound to hemoglobin. And then the x-axis represents the partial pressure. What is the partial pressure of oxygen? So let's say here no oxygen is bound to hemoglobin, and then on the middle here, that is where half of the hemoglobin has bound to oxygen, like 50%. Okay, so we call this as P50, where the partial pressure of oxygen is 50%. And then by the time you're reaching the top here, that's where you are nearly reaching 100% saturation. Okay, that's what happens. So this is the normal. But once the curve, so what happens is this. Let's say when we are looking at the P50, okay, that is how what should be the partial pressure of carbon or the partial pressure of oxygen for 50% of hemoglobin to be occupied by oxygen. So let's say this value here is 20. Okay, let's just take that 20 as just a value. So what happens is that if this graph goes in this direction, we call that as a right shift. And look what is going to happen. So for the right shift, the graph might go like that. Like that. So if you want to look again at the same thing, the P50, the partial pressure of oxygen that is going to bind to half of the hemoglobin. Let's say the value there is going to be a 40. So you see that for you to have half of the hemoglobin bound by the oxygen, you now need a higher partial pressure. Okay? What does that mean? It means that it's now very hard for oxygen to bind to the hemoglobin because you now need a lot of oxygen or a higher partial pressure of oxygen to bind to the hemoglobin. So a right shift of the oxygen saturation of hemoglobin means that it is very hard for oxygen to bind to the hemoglobin. What makes it hard for oxygen to bind to hemoglobin? It's these factors that you can see here, these ones that we've listed here. And we actually call this as the Bohr effect. This effect basically promotes because it's very hard for oxygen to be bound to hemoglobin. It promotes the release of oxygen from the hemoglobin so that the oxygen can go and bind to the tissues. If the opposite happens where you move in this direction, we call that as the Howden effect. And that means that these factors that I've listed here have basically decreased. Okay, now to answer our question, what is meant by the shift of oxygen dissociation to the right? So that is basically a shift of oxygen dissociation to the right indicates that hemoglobin has a decreased affinity for oxygen. If it is decreased affinity, it means now that the oxygen can actually be released, okay, into the tissues. All right, it can go and bind to the tissue. That means that a given partial pressure of oxygen, hemoglobin will be less saturated. Okay, so it's going to be more likely to be released. In other words, at a specific partial pressure, if the graph shifts to the right, it means that that same partial pressure will not be able to bind more to the hemoglobin. So it promotes the oxygen to be released. And then the second part was saying enumerate the causes of such a shift. We've said an increase in the partial pressure of carbon dioxide, acidosis, which is a decrease in pH or an increase in hydrogen, and then you have got 2,3 bisphosphoglycerate. It used to be called the diphosphoglycerate, but you don't call it di. It's 2,3 bisphosphoglycerate. And then also exercise, because this will increase the temperature and of course increase in temperature. And then the other part of the question was saying, is this shift advantageous? Yes, this shift is advantageous. Why? Because think of in exercise, you need a lot of oxygen to enter into the cells. Okay, into the cells. So you need to the oxygen to have a low affinity to hemoglobin so that it can be released and delivered to the cells for cellular respiration to happen. I hope that is okay. Does that make sense? Okay, very good. Number 4 is saying a patient has red blood cell count 4 million per cubic millimeter, hemoglobin content 8 gram per deciliter, the packed cell volume is 30%. What kind of anemia is this person suffering from? Show calculations of the various blood indices. Okay, so the blood indices that we have got MCV. MCV just tells you the average volume of a red blood cell. Okay, one red blood cell, like what's the volume of one red blood cell? And the way you calculate the MCV, mean corpuscular hemoglobin, is you get the packed cell volume, the percentage of packed cell volume, you divide by the red blood cells. Please, this value that you use is already in 10 to the power 6. Okay, so that's what you need to know. It is already in this unit. So you don't include it when you are calculating. So the packed cell volume in this case, actually, you don't forget to multiply by a 10. The packed cell volume here you can see is the 30. You multiply by that by 10, and then you divide by the red blood cells, the red blood cell count. So here it is been given as 4 million per cubic decimeter. Million means 4 times 10 to the power 6. Okay, per cubic decimeter. So that is mm cubed. Okay, so this one is already in 10 to the power 6. So you just write the 4 down. Don't include the 10 to the power 6 because it's actually already considered part of the formula. And that is going to give you 75. Now the mean corpuscular volume, the normal value is between 80 and 900. The unit is in femtoliters. So 75 is below 80. And mean corpuscular hemoglobin tells you the size of the red cells. So this means that this is a small cell because the value is smaller than normal. So that is a microcytic red blood cell. The other one that want to calculate another blood index is MCH. That's the mean corpuscular hemoglobin. Now, the mean corpuscular hemoglobin is the measure of the average amount of hemoglobin per red cell. Like if you have got one red cell, what is the average hemoglobin that you have there in one red cell? Okay. And this one again is found by HB times 10 over red blood cells. You don't use the packed cell volume, you use the HB. In this case, the HB was 8. And then multiply by 10 divided by 4, which is the red blood cell count. Now for MCH, the answer is in picograms. It is in grams. The normal value is from 27 to 31. This value is 20, so that is low. MCH represents the amount of hemoglobin. And hemoglobin is actually what is responsible for the color of the cells. Okay? So since it is low, it means that this is actually for color, we use chromic. So this is hypochromic. Okay? Hypochromic. So this is actually going to be a microcytic hypochromic anemia. But these are not the only indices. We also have what we call MCHC, the mean corpuscular hemoglobin concentration. Remember we said that MCV is the volume of one red cell. But if you take the average volume of all the red cells, okay, that's now called MCHC. mean corpuscular hemoglobin concentration, the measure of the concentration of hemoglobin in a given volume. Actually, this is the concentration of hemoglobin in a given volume. The MCV was just the measure of the average amount of hemoglobin in one red cell. Like how much hemoglobin do you have in one red cell? Now, if you are looking at how much hemoglobin do you have in a packed in a volume, in a certain volume, not in one red cell, but like you've gotten at least a good sample of blood, and then in all of them, how much hemoglobin do you have, the average? And this one is given by HB, that is the hemoglobin, times 100 over PCV, the packed cell volume. HB here is 8 times 100 over the packed cell volume is 30. That will give you 26. MCHC, the normal value is from 32 to 36. Okay. Now, MCHC, because it's also talking about hemoglobin, it also talks about the color. So that is going to be, because the 26 is less than 32, that's also going to be hypochromia. Like the color is going to be low color. So this is again confirming that this is hypochromic, microcytic hypochromic. Another blood index, of course, is normally used, but it's also part of them, is the color index. Color index is found by the hemoglobin. So color index is the percentage of hemoglobin divided by the percentage of the red cells, okay, times 100%. Now, for you to find the percentage of hemoglobin, you use the hemoglobin that you've been given, and then now you use the expected normal value of hemoglobin for a certain gender. Here, the HB that you've been given is 8. For males, they have got an expected HB. For females, they also have got an expected HB. Okay. So in this case, we have just used that the standard HB is 14.8. Actually, for females, the accepted value of the hemoglobin is lower than males, because females are always losing blood, so a low value is accepted. So percentage HB, therefore, we use the HB for this patient, which is 8, and then you use the standard one. Okay, in this case, we are taking that the standard is 14.8. For males, we accept 14. For females, we accept even HB of 12. Okay. And then the red blood cells also, it's a percentage that we're calculating. So the percentage, you use what you have been given. Okay. In this case, it's 4. And then the normal red blood cell count is supposed to be, in this case, we take 5 million. Okay. As the normal accepted value. Once you do this, you are going to multiply by 100. The normal value is 0.85 to 1.2. Normally we get 1, like 0.85 to 1. So you can see 0.68 is more. So if the color index is low, that tells you that actually that's anemia. And the color is low. So everything all confirms here. But normally if they ask you to calculate color in the blood indices, you calculate MCV, MCH, and MCHC. Okay, so this is microcytic uh, ma- hypochromic anemia. Number 5 is saying explain the phenomenon of excitation contraction coupling in cardiac muscles. Okay, so in other words, what's the mechanism of contraction in a cardiac muscle? Now, contraction all starts with what we call an action potential. Okay, so when you have got a muscle, you have got this region here, we call this as a T tubule. A T tubule. And then you can have either a nerve, it can be a nerve. Let's say these are the dendrites of a nerve. Okay. So for this nerve, action potential is going to start from the body. This is the body. Action potential is going to spread. Once that action potential gets to spread, is going to come here. These nerves, we call them the presynaptic neurons, because they are before a synapse. Presynaptic. They are going to release a neurotransmitter. That neurotransmitter is going to come and bind on the postsynaptic cells. And then an action potential will still be generated on this cell. In other words, action potential, wherever it has come from, it is going to spread via a T tubule. Once that action potential gets to spread, via this T tubule. The T tubules contain what we call the L type calcium channels. This is, remember, for a cardiac muscle. So the L type calcium channels. Okay. So the depolarization, because action potential actually what it does is that it basically spreads what we call depolarization. The inside of the cell becoming more negative. That's the meaning of depolarization. So what happens in that case is that the L type calcium channels are going to open. And what does that cause? Once these calcium channels open, it will cause now calcium to start moving into the cell. Okay, a small amount of calcium is going to enter the cell from the extracellular fluid from the outside. Now, once that happens, inside this cell, you have got a smooth endoplasmic reticulum of a cell, of a skeletal muscle, of a muscle. Now, the smooth endoplasmic reticulum of a muscle, we call it the sarcoplasmic reticulum. So what happens that once this calcium enters the cell, it's going to come and bind. There is a receptor here, which we call the iodine receptor, which is this one. Once this calcium, only a small amount of calcium that came from outside the cell, binds on the iodine receptor, it is going to cause the opening of the sarcoplasmic reticulum because this guy actually stores calcium. So now a lot of calcium is now going to move out and go into the intracellular fluid or the cytosol. So we call this a CICR, calcium induced calcium release, or calcium in, calcium out. Okay? And once you have got a lot of this calcium now that has entered the intracellular fluid, you actually have a structure here. Muscles basically contract because of what we call the thick filaments and the thin filaments. The thick filament is the myosin. This is the myosin. Okay, that's the thick filament. And then you have got the thin filaments. The thin filaments are actually a lot. You have got the commonest one is actin. Actin is a guy which looks like that. It looks like so. Okay. Besides actin being a thin filament, you also have what we call troponin and tropomyosin. Troponin, you have got troponin C, troponin I, and then troponin. Uh, so you have got C for calcium, and then you have got I, and then which other one? Okay, I'll remember the third one. Okay, so that is what happened, and then you have got tropomyosin. So once the calcium comes in in this one, okay, once the calcium comes here, it is going to cause now the calcium to go and bind on tropomyosin. Actually, this actin that you can see here has got a head. Myosin has got a head actually. Okay, it has got a head that binds to calcium to actin. But now the tropomyosin is going to close the heads. Okay, it closes the heads on the actin so that it closes the binding site on the actin where the head from the myosin is supposed to bind. So actin has got what we call the myosin binding site. That is where the head of the myosin binds. And that head, that site where this head binds is closed by the tropomyosin. Now, to attach to the tropomyosin, you have got three types of troponins. Three types. You have got what we call, it's like this. There is troponin I, troponin T, and troponin C. I is basically the inhibitor. T is represent the troponin T is basically representing tropomyosin. It binds to tropomyosin. And then the troponin C, that's the one that binds calcium. So once this calcium has been released from the sarcoplasmic reticulum, it's going to come and bind to the troponin C. Once it binds to troponin C, it is going to now remove the inhibitor, which is the troponin I. Okay, and that will cause now the the troponin T, of course, which is binding to tropomyosin. It will pull the tropomyosin. Remember, all these three types of troponin are bound to the tropomyosin. Okay, so calcium comes to bind to troponin C. Once it does bind to troponin C, what happens at that particular time is that it will cause now the tropomyosin to come out of the site here where the head of the tropomyosin is supposed to bind. And so what happens since the tropomyosin has gone out, so it is now out, okay, like that, the myosin head can now bind to the binding site on the actin. And then what happens once it binds, this requires ATP. Once it binds, it's going to now move in that direction. It will pull it. And that's how a contraction happens. And we call those as so the first thing that happens is the cross bridge cycling. Okay. The head of the myosin binds to the myosin binding site on the actin. And then it is pulled. And that's how the contraction is going to happen. Once this is done, contraction has happened, relaxation is supposed to happen. Relaxation also requires ATP. So what happens that for relaxation to happen, you just basically need to take away this calcium. So there are too many mechanisms by which that can happen. Number one is that the calcium can go back into the sarcoplasmic reticulum using an exchanger which we call a SECA. That is the sarcoplasm endoplasmic reticulum calcium ATPase pump. So that is going to push calcium back into the sarcoplasmic reticulum. Okay. And then another mechanism is using the sodium calcium exchanger. And that is the calcium is now going to be removed from ICF back to the ECF. So what will happen is that the calcium will be moving out while the sodium is going to be entering in. So that is what happens. Okay. One important thing about cardiac muscle contraction is that contraction is going to be graded. In other words, the muscle can contract a bit, and then another small contraction can happen, another small contraction can happen, and then all these contractions can add up to give you a bigger contraction. That doesn't happen in skeletal muscles because in skeletal muscles we have got what we call the one or none principle. It's either the muscle contracts or it doesn't contract at all. There's nothing like small contractions will add up to give you a big contraction. That doesn't happen. All right. So that is important to understand. Okay, very, very important. And take note that in skeletal cardiac muscle, you need calcium from the outside of the cell. And because cardiac muscles have got gap junctions, if one cardiac muscle contracts, it action potential can spread to the next cardiac muscle and then also contract, and we call that as a syncytium. Does that make sense now? Is that okay? Okay. So now just a quick summary of everything that I've talked about. The first thing is that you have got an action potential. That action potential is going to enter the cell. Okay, like the here again a simple description so that you just put everything all together, because it's actually very important for you to understand the different mechanisms of contraction in the three types of muscles. They are not the same. Okay, so you have got an action potential wherever it is going to come from. It can come from a nerve, it can come just from excitation of the muscle itself. So the action potential is going to spread via a T-tubule. And once that action potential reaches where the T-tubule is near in the T-tubule, there's depolarization that happens here. And there is what we call the L-type calcium channel. Because of this action potential that has reached here, a depolarization has happened. Calcium will move from outside the cell to the inside of the cell. Once this calcium enters the inside of the cell, it is going to go and bind to what we call the rhodine receptor. Once it binds the rhodine receptor to cause the sarcoplasmic reticulum to open, and then calcium is going to be released from outside the sarcoplasmic reticulum. And we call this mechanism as calcium in, calcium out. So calcium into the cell, calcium outside, out of the sarcoplasmic reticulum. And what is of importance is now this calcium that is here. So you have got filaments that cause contraction in muscles, basically all types of muscles. We have got the thin filaments, and then we have got the thick filaments. Thick filament is basically myosin. And then the thin filaments, you have got three. You have got the main one, which is actin, and then there is a myosin, and then there is a troponin. Troponin is of three types. There is troponin I, troponin C, and troponin T. Okay, so normally this is this one that I've drawn here. This is the myosin. It has got the head. Okay, myosin head. It actually has two heads, but I'll just show one. Normally, this myosin binds to actin. So actin has got a site for the binding of the head. This is actin. Now, when contraction is not happening, the site where the head of myosin binds on actin is covered, and it's actually covered by what we call tropomyosin. So the tropomyosin also attached to it is the troponin, the three types of troponin. This is troponin. So this calcium that came into the cell, once it's there, is now going to bind on the troponin. Specifically, the troponin has got I, C, and T to come and bind on the troponin C. C is to mean calcium. Once the calcium binds on the troponin C, the troponin is like going to pull away the tropomyosin away from the actin. So the tropomyosin has been pulled away from the actin. So now the actin, the binding site for the head of the myosin is now free. So it can now bind, and once it binds, it will now like pull back. Okay, the head is going of the myosin is going to pull back, and that's how a contraction gets to happen. Does that make sense now? Okay, please feel free in case you are still not okay. All right, there's no rush. So this same binding of the head to the binding site and then pulling back, we call that as a cross bridge cycling, and that's how contraction happens. So you also need to know how relaxation happens. Okay, calcium will either go back into the sarcoplasmic reticulum using Seca pump, or to use the calcium sodium transport. The difference with the contraction in smooth muscles is this, and you need to know, please, what is the difference? How do smooth muscles contract? Because that was a cardiac muscle. You still have what we call an action potential. Okay, so but smooth muscles are controlled by the autonomic nervous system. They are involuntary. You don't say, okay, stomach contract, relax. Okay, intestine contract. No, you don't do that because it is involuntary. So it is controlled by the autonomic nervous system, the hormones, and also the stretch receptors. So what happens is that once these guys are controlled like this, okay, action potential is going to spread. So there are different things. The important thing when it comes to contraction is basically calcium. Okay. So again, this is the cell which contains the myosin and the actins inside, which are going to contract. But the thing is that you need calcium for those guys to contract. For smooth muscles, where does the calcium come from? You need to know this. Three main ways by which the calcium can come in. It can be from a depolarization. So the calcium is going to, so depolarization is going to come in, and then that depolarization is going to open the calcium channels here. Okay. And these are voltage gated. Like when there's a change in voltage, and then the calcium gets to enter the cell. Another way by which calcium can come in is through the hormones and neurotransmitters. When an hormone or neurotransmitter binds on this cell, which is a smooth muscle, it is going to now cause the release of calcium. Okay, actually, it will not cause the release of calcium. Instead, it will cause the opening of calcium channels. We call those as ligand-gated calcium channels because they only open when a ligand binds, and a ligand includes either a hormone or neurotransmitter. Okay. So by these two mechanisms, either by action potential depolarization happens, and then there's opening of the voltage gated calcium channels or a hormone. There's the opening of the ligand gated calcium channel. Once that calcium enters the cell, it is also going to stimulate the release of calcium from the sarcoplasmic reticulum, which is inside here. Okay, so little calcium has entered, and then more calcium is released from the sarcoplasmic reticulum. So that is calcium in, calcium out. That is the first two mechanisms. Another mechanism by which it can happen is that hormones, neurotransmitter, or even hormones can actually work via what we call a G-coupled protein, and that G-coupled protein at the end of it, or there's going to be the activation of the G-coupled protein that is going to cleave, is going to break down PIP3. Okay, when this PIP3 is broken down, there's going to be production of what we call IP3, and then also DAG, diacylglycerol. And once this happens, the IP3, when it increases, this is inositol triphosphate. It is now going to cause the calcium, okay, because it's actually happening inside the cell. It's going to cause now the calcium to be released from the sarcoplasmic reticulum, and that still increases the calcium. And then the calcium is now, this is the difference with the other muscle that we talked about. When the calcium increases here inside the cell, it does not go and bind to the troponin C, because in smooth muscles you don't have that troponin C. Okay, you don't have the troponin. Instead, there is another guy which you call calmodulin. So this calcium is going to come and bind to calmodulin. And what calmodulin does is that calmodulin is going to lead to the formation of something, activation of something. Okay, so calmodulin is going to, so the calcium that is bound to calmodulin forms the calcium calmodulin complex, and that is going to activate what we call the myosin light chain kinase. Okay, so let's say this is a calcium, this is a calmodulin. These guys are going to go and activate the myosin light chain kinase. Once the myosin light chain kinase is activated, this guy phosphorylates a myosin. Okay, this is a myosin. This is a myosin. So this guy is going to get a phosphate and go and add it on the myosin, and that's called phosphorylation. And once the myosin has got that phosphate, okay, once the myosin has got that phosphate, the myosin head can now bind to the actin. So what causes the binding of the myosin head to the actin is the phosphorylation of the myosin. Specifically, remember this was the myosin light chain kinase. A kinase is an enzyme that has a phosphate. So this kinase is adding a phosphate to what? It's adding a phosphate to the myosin light chain because it's a myosin light chain kinase. Okay, so it is myosin light chain. Once a phosphate is added, then it can bind to actin. And then those crossbreed cycling are going to happen that causes contraction. And the way smooth muscles relax is that the calcium is going to be removed by pumps or exchangers. Okay, or the myosin light chain is now, you are going to have an enzyme which we call the myosin light chain phosphatase. A phosphatase removes calcium, removes a phosphate. Once the calcium is removed from the cell, that phosphate will now be, which was added to the myosin light chain, will be removed using the enzyme phosphatase. And because the phosphorylation has happened, relaxation is going to happen. Take note that smooth muscles don't contain troponin. Okay, contraction is also slow. So that is what happens. And the last one for to understand is the skeletal muscle, how it contracts. Skeletal muscle is very similar to cardiac muscle in that, but one thing is that for skeletal muscle, you have got this. Remember, for a smooth muscle, we said this is the T tubule, and then we said that you have got the L type calcium channel, but that thing is not there. Instead, we have got what we call the dihydropyridine receptor. Okay, this one here. This is the T tube. So when action potential comes and reaches here, there's going to be this depolarization that is going to open this dihydropyridine receptor, which is voltage gated. The dihydropyridine receptor is actually coupled to the radon receptor. The radon receptor is present. Okay, these are coupled. This guy, the radon receptor, is present on the sarcoplasmic reticulum. So once the dihydropyridine receptor is activated, it is going to cause, okay, is going to cause the iodine receptor to be activated, and then calcium is going to be removed. So what happens first is action potential reaches the cell, the T tubules, and that activates the dihydropyridine receptors, which are coupled to iodine receptors, and that will cause now calcium to leave the cell. Okay, and then the calcium can go and bind to the troponin C, just like in cardiac muscle. Everything else is the same. It's just that here the calcium in, calcium out does not exist. Okay, and that is going to cause contraction. If relaxation is to happen, you also have a second pump here, just like in cardiac muscles. The calcium should be taken back into the cell. Okay, but then for small skeletal muscles, there is no that calcium sodium exchanger which was removing the calcium outside, because there's no calcium that came from the outside here. So that is what happens, and this is the mechanism that you can see here. Okay, depolarization, T tubule, in the T tubules, opening of full calcium release channels are the radon receptors, which are attached to the dihydropyridine receptors. Calcium inside the cell increases, it binds to troponin C. Troponin C will cause the tropomyosin to be removed from the actin active site, and that will bind the myosin head to the actin, and that forms the cross bridge cycling, and that causes contraction. Okay, so that is what happens. Does this make sense? Okay, very good. Now, number 6 is saying state and define normal value of the following: air, alveolar air, tidal volume, total lung volume, residue volume, respiratory rate. Okay, the first thing that I want you to understand is what happens when you are breathing in and out. So, when you are breathing in and out, okay, the amount of air, like right now, you are breathing in and out, even if you are not even aware. So the normal amount of air that you breathe in and out, when you are resting, we call that as the tidal volume. So you breathe in, you breathe out. So going up is in, going down is out. So from this top to the bottom here, we call that as the tidal volume, and it's about 500 mils. Okay, 500 mils. That's the normal that we breathe in and out. Please take note, because that's important. Now, sometimes you can forcibly breathe in, like you can forcibly breathe in. So you can take in a very deep breath. You can see how you go up. And then you can also forcibly breathe out, forcibly. Okay, like that. So the amount of air that you are going to forcibly breathe in, we call that as the inspiratory reserve volume. Inspiratory reserve volume. And then the amount of air that you can get to forcibly breathe out. Now, this is the trick thing. Okay, before I talk about that, the amount of air that you can forcefully breathe out, that's known as the expiratory reserve volume. The thing is this: even if you forcibly breathe out, there is always some air that remain in the lungs. Not all the air is going to be removed, and we call that as a reserve volume. Okay, so these are the volumes. These are the volumes you need to know: the tidal volume, inspiratory reserve volume. The reserve volumes are not a combination. Okay, so inspiratory reserve volume is just the amount of air that you are going to forcibly breathe in, excluding that part that you normally breathe in and out. And then expiratory reserve volume is the amount of air that you can forcefully breathe out, besides the normal that you can just breathe out. Okay, and then you have got the reserve volume. Now, when you start combining volumes, that gives you what we call capacities. For example, if you start from the beginning, okay, you start from the very beginning, like you combine the tidal volume with inspiratory reserve volume. So, meaning that from here at this point all the way to the maximum point. So, what have we combined? The tidal volume and the inspiratory reserve volume. When you combine volumes, you form capacities. So the combination of the tidal volume and the inspiratory reserve volume, that forms what we call the inspiratory capacity. Again, you can combine the inspiratory volume, okay, with the expiratory reserve volume. But in that case, you're only looking at the part that you actually breathe out. So you are starting from here all the way to the very, very bottom. You can see that. So basically, for this one, you're combining the expiratory reserve volume with the reserve volume. Okay? Expiratory reserve volume with the reserve volume, and that will give you what we call the functional reserve capacity. Remember, these are capacities. Okay? So functional reserve capacity is a combination of expiratory reserve volume with the residue capacity, the amount of air that remains in the lungs. Now, if you start from the volume, the amount of air that you forcibly breathe in and then forcibly breathe out all together, forcibly breathe in and forcibly breathe out, we call that as a vital capacity. It's a capacity. So for the vital capacity, you basically combine, there is the inspiratory reserve volume, there is a tidal volume, and then there is the expiratory reserve volume. That's a vital capacity. And then when you take now the total amount of air that is present in the lungs, from the very bottom to the very top, we call that as the lung capacity. Okay, so those are the things. So the alveolar air is basically the amount of air that is going to be in the lungs for gaseous exchange. Because when you are breathing in, not all the air, look at this, the respiratory tract is like this. You have got the trachea, the primary principal bronchioles, the secondary ones, and then you are going to have the tertiary and such. Now, gaseous exchange happens at the end where you have got the alveolar, meaning that air that is actually here in this region is not involved in gaseous exchange. So we call that as a dead space. That air is not used in gaseous exchange because the air that is used in gaseous exchange is the one that is here. So alveolar air is basically the air that is present in the alveoli for gaseous exchange. Okay, it's the amount of fresh air that enters the alveoli with each breath, and then you remove the dead space. So the amount of air that enters the alveolar with each breath, we call that as the tidal volume. And then remove the dead space, the amount of air that is not used for what? The amount of air that is not used for gaseous exchange. So the tidal volume we had said that is 500. That's the amount of air that enters the body. And then the dead space is about 150. That's the amount of air that is not involved in gaseous exchange. So that leaves you with the alveolar air being 350 mils. Remember the question is state and define the normal values of the following. So you state and then define the normal value. Okay. The next one is the tidal volume. We already said that the tidal volume is the amount of inspired or expired air during normal quiet breathing, and it's about 500 mils in an average adult. And then the next one is the total lung volume. Okay, so the total lung volume, we actually don't call it volume, it's the total lung capacity because it's a combination of volumes. So it's the maximum volume of air the lungs can hold after a maximum inspiration, the maximum amount of air that can be held, and that's about 6 liters or 6,000 mils. And then residual volume is the volume of air that remains in the lungs after maximal expiration, like after you expire maximally, and that's about 1.2 liters or 1,200 mils. Okay, respiratory rate is the number of breaths that we take per minute. Like right now, if you count, okay, you'll be looking at how someone is breathing. So how many breaths do you take in a minute? That's one. That's two. Three. So in a minute, the normal is between 20, 12 to 20 breaths per minute in adults. Okay, so those are the terms. Number seven was saying, okay, before we go to seven, is number six okay?
Yes, it is. So, can they even ask you to draw the diagram?
Yes, they can. They can ask you to draw it. Okay. So so describe the function of the juxtaglomerular complex. Well, the juxtaglomerular complex is like this. Remember the when you see the way the lungs are, you have got what you call a nephron, which is a functional unit of the kidney. A nephron is going to be made of different parts. So here you have got what we call the afferent arteriole. This afferent arteriole, by the time it reaches out a structure like this, it will go like that, and then it will come out as the efferent arteriole. Okay, this is an arteriole, and then you are going to have a structure here like that. We call this as the Bowman's capsule, and then this is going to go down. This first part is going to be called the proximal convoluted tubule. This guy here is the Bowman's capsule. Okay, proximal convoluted tubule. This guy is going to become what we call the loop of Henle. It has got the part that is going down, we call that as a descending loop of Henle. There's a part that goes up, we call that as the ascending loop of Henle. And then there's going to be the distal convoluted tubule. We call it convoluted because it's not straight like this, it actually looks like that. Okay, that's why it's called convoluted. But now the distal convoluted tubule, what it does is that it goes like this, like that. So there is a part of the distal convoluted tubule that rests on the, so this part of the arteriole, we call it the afferent. Alpha A is meaning towards. So this is going towards the Bowman's capsule. And then this part of the arteriole, we call it the efferent. E means away, efferent arteriole. And then this blood vessel itself here, we call it the glomerulus. Glomerulus. So this is basically a tuft of capillaries. Okay. As you can see the way I've drawn it, the afferent arteriole is big in diameter, the efferent is small in diameter. And that's important. So the part of the distal convoluted tubule that rests on the afferent arteriole is going to have cells, and we call those cells. So the afferent arteriole, the diameter is wide, it's very big, so that a lot of blood can be going to the glomerulus. But the efferent, it is small, so that when a lot of blood has reached the glomerulus, it shouldn't quickly leave. Okay, so you have got a big inlet but a small outlet, so that a lot comes in, but it is not leaving out very fast. All right. So I was saying this is the distal convoluted tubule. Now the cells of the distal convoluted tubule that rests on the afferent arteriole, we call those as the macula densa. And this is the cell that you can see here. This is the macula densa. Okay, macula densa. And then now the afferent arteriole itself has got epithelial cells. Remember, each artery has got an epithelial lining, and we call those as the squamous cells. Specifically for arteries, we call for blood vessels, we call them the endothelium. So these endothelial cells, or the afferent arterioles, we call them the juxtaglomerular cells. So you have got the juxtaglomerular cells, you have got the macular denser cells, which are cells of the distal convoluted tubule that rests on the afferent arteriole, and then you have got these cells in between the two. We call those as the lattice cells or mesangial cells or network cells. So these three things make up what we call the juxtaglomerular apparatus. Juxtaglomerular apparatus. Now, the question is asking to say, describe the function of the juxtaglomerular complex. Okay? So this is the complex made up of three things. When you look at the macular denser cells, these are cells of the distal convoluted tubule. So what happens is that when filtration happens here, okay, salt is going to be filtered, that is sodium. Now imagine a lot of sodium has been filtered. Okay, a lot of sodium has been filtered. Well, if a lot of sodium has been filtered, it means that a lot of sodium is actually going to go up and a lot of sodium is going to reach the distal convoluted tubule. And if a lot of sodium reaches the distal convoluted tubule, that is going to stimulate the macular denser cells. Okay, so what is stimulating macular denser cells? Number one is a high osmolarity. or high sodium and chloride levels. Because if you have got a lot of salt, it means that the concentration is going to be high. So this corresponds to high plasma osmolarity, because the only way you can have a lot of sodium in urine is because you have got a lot of sodium in blood. That's one stimuli. So what happens is that when you have got high plasma levels of sodium, it is going to go and stimulate the macular denser cells. Once the macular denser cells are stimulated, something happens at that particular time. Okay, these guys are going to stimulate what we call the RAS system. But number one is the first thing is what happens is that there's going to be the stimulation of the sympathetic nervous system. Okay, that is what is going to be stimulated. When the sympathetic nervous system is stimulated, the aim is to try and come and reduce this concentration of the sodium. But take note of this: when you have got high levels, what stimulates the macular denser cells? Okay, this pen. Okay, all right. So for the macular denser cells, what stimulates them is high levels of sodium. And what happens that if since you have got a lot of sodium in blood, you want to dilute that sodium. How can you dilute it? You want to make sure that you are not producing a lot of urine. You are keeping that water to be in the blood vessel so that the sodium can be diluted. So high levels of sodium chloride is going to stimulate the macular denser, and then these guys are going to release adenosine. Adenosine causes relaxation. It is a vaso. Actually, it doesn't cause relaxation, but it causes the constriction of the afferent arteries, those arteries that are taking blood to the kidney. Why is it doing so? Imagine if this guy becomes smaller. It means that a lot of sodium will not be able to reach the kidney. Okay, so that is what happens to prevent that high levels of sodium reaching the kidney. So actually reduces the renal blood flow, and that decreases the glomerular filtration rate. The opposite is going to be true. When you have got low levels of sodium chloride, it means that only a little sodium chloride is going to reach the macular denser. And so if little reaches the macular denser, it stimulates the release of the prostaglandins and nitric oxide. Now, these guys are vasodilators. They are going to cause the blood vessels, specifically the afferent arteriole, to dilate. If the afferent arteriole dilates, what happens in that case? It means that a lot of blood, isn't it? A lot of blood is supposed to get to reach to the kidney. Okay? But then the opposite of thing is going to happen in this case. When you have got low levels of sodium chloride, what happens is that, of course, dilatation has happened, okay, of the afferent arteriole. And then that is also going to lead to the release of renin. And what these do is that instead of the nitric oxide and prostaglandin just dilating the vessels, the release of renin is going to preferentially constrict the instead of, remember, you have got the afferent, and then you have got the Bowman's capsule, and then you have got the efferent. So instead of this one only dilating alone, this guy here is going to constrict more, so that a lot of blood is actually coming here, but a lot of blood is leaving. And what happens in that case is that you are bringing now a lot of salt to the kidney, so that will increase glomerular filtration rate. Okay, that is going to increase the glomerular filtration rate. So that is what happens. In other words, low levels of sodium chloride increases the glomerular filtration rate, while the high levels of sodium chloride will reduce the glomerular filtration rate. That's what the macular denser cells do. The mesangial cells, which is another component, also known as the Lassie cells or network cells or poikisen cells, these ones secrete what we call erythropoietin. And this guy is involved in the production of the red blood cells. Okay, red blood cells. And then now what remains is, what do the JG cells do? The juxtaglomerular cells. What the juxtaglomerular cells do is that they also respond to three things mainly. Number one, they'll respond to blood pressure. Okay, they'll respond to blood pressure. Number two, they will be able to respond to the levels of sodium. Number three, to sympathetic stimulation. So what happens is this: again, If you have got a low blood pressure in the body, okay, low blood pressure in the body, low BP, it means that only little blood is going to reach the kidney. Okay, little blood reaches the kidney. Because of that little blood reaching the kidney, even the filtration is going to be small. So specifically the afferent arterioles, remember that's where the juxtaglomeralar cells are. The afferent arterioles have got baroreceptors. These are cells that sense pressure. Baro is pressure. They'll sense the pressure, and since the pressure is decreased in this case, what that does is that is going to stimulate something. But before I talk about what it stimulates, also when you have got low levels of sodium chloride, low levels of these guys is also going to stimulate the macular denser cells, as we said earlier, all right? And then the sympathetic stimulation can also happen via the beta 1 adrenergic receptors directly. But what happens is that with all these stimuli, what happens is it leads to the secretion. The juxtaglomerular cells are going to release what we call renin. So when does renin get released? When there is low BP, okay? Look at this. Low BP basically means that you have got low volume, okay? You have got low volume. Another stimulus is that if you have got high levels of sodium, a lot of salt. Because if you have got a lot of, when you have got a low BP, you want to come and retain water, not release the water in the urine so that it still remains in the blood vessels. That's number one. Number two, when you have got high levels of sodium, you still don't want to form a lot of urine. You want to maintain the water so that it goes and dilutes that urine. Okay? That's number two thing that happens. So either low levels of blood volume or high concentration of sodium, that is uh, an hyperosmolar, okay, or a high concentrated blood. This will stimulate the release of renin from the JG cells. Now, what renin does is this. Renin is the liver produces an enzyme which we call and a hormone called angiotensinogen. So this renin is now going to convert that angiotensinogen into angiotensin 1. The lungs will produce what we call angiotensin converting enzyme, which is going to now come and convert angiotensin 1 into angiotensin 2, which is a potent vasoconstrictor. Okay, this angiotensin 2, it can do many things. It can cause the release of ADH from the posterior pituitary gland, which will go and stimulate the collecting ducts to reabsorb water. If water is reabsorbed, it means that you are going to now increase the BP. You're also going to dilute the sodium so that the high levels of sodium cannot be high like that. Okay. Number two that angiotensin 2 does is that it can vasoconstrict afferent arterioles, because you remember it's a potent vasoconstrictor. If it vasoconstricts them, it means that the glomerular filtration rate is going to reduce, because the blood vessel that was bringing blood to the kidney has been reduced in size. Okay. So it reduces glomerular filtration rate and renal blood flow. And that means that you are now going to maintain the little water that you have will not go to the kidney to form urine, it will still be in blood. And then angiotensin 2 can also stimulate the hypothalamus to cause thirsty so that you can drink a lot of water, because remember you have got a low BP. Angiotensin 2 can also stimulate the adrenal gland. The adrenal gland has a cortex and a medulla. The cortex has got the zona glomerulosa, zona fasciculata, zona reticularis, specifically the zona glomerulosa, which releases aldosterone. And aldosterone promotes the reabsorption of salt. Okay, and the water. So we are interested in actually the water reabsorption, because if you reabsorb the salt is already high in this case. Okay, so this is what happens. This is the juxtaglomerular complex. Does that make sense? Okay, very good. Number 8 is saying, based on your knowledge of muscle physiology, what is the function of rhodon receptors? What is the rationale of using dantrolene, which is a rhodon receptor antagonist in the treatment of malignant hyperthermia? Now, we already explained what rhodon receptors do, isn't it, in details? We said that if you have got the sarcoplasmic reticulum in muscles, which stores calcium, okay? This guy stores calcium. And then you have got a channel here, a channel which we call the rhodine receptor. So rhodine receptor are calcium releasing channels on the sarcoplasmic reticulum of muscle cells. And the question is saying, based on your knowledge, what is the function? So this guy, the function is that it is going to be able to release calcium in muscles, and once that calcium is released, it's going to cause, number one, in skeletal muscles we actually have rhodine receptor 1, in cardiac muscle rhodine receptor 2. Okay, these guys, rhodine receptor 1 is attached to the hydropyridine receptor, as we said. Rhodine receptor 2, this guy works by calcium in, calcium out, and it's related to the L-type calcium channels. And what the calcium do is that the calcium is going to be released from this sarcoplasmic reticulum into the cytoplasm. The calcium binds to the troponin C, or it can activate the calmodulin, uh, myosin light chain kinase complex. Okay, and that still causes contraction. So basically, rhodine receptor is a gatekeeper for calcium release to cause contraction. Okay, so that is what it does. So we've talked about the role. The second part is saying, what is the rationale of using dantrolene in treatment of malignant hypertension? Now, remember, dantrolene is a rhodine receptor antagonist. Antagonist means that once it binds on the rhodine receptor, it will cause the rhodine receptor to fail to release calcium, so the calcium will not be released. Okay, so the first thing is you need to understand what malignant hyperthermia itself is. Malignant hyperthermia is basically heat. Okay, like high levels. Hyper means high. So malignant hyperthermia is a life-threatening genetic disorder where there is a gain of function mutation. So there is a mutation in the rhodine receptor which makes it to gain function, meaning that it ends up releasing a lot of calcium. Okay, that's a problem with this condition. And this specifically happens when you expose a person to anesthesia. This is used during surgery, for example, halothane or even succinylcholine. Okay, so these guys will cause a mutation to the receptor, and then they become hyperactive. So how it happens is that there's a mutation which causes uncontrolled calcium release. So a lot of calcium is going to be released from here using this rhodine receptor. So the calcium is being released and being released and being released. And what that calcium does is that it will cause sustained muscle contraction, massive ATP consumption, because muscle contraction uses ATP, and it also causes the generation of heat, and also lactic acidosis and hyperkalemia. These are the signs and symptoms of someone that has got this malignant hyperthermia. So there's going to be hyperthermia, which is heat, muscle rigidity because of sustained contraction, tachycardia also, and metabolic acidosis because of this lactic acid that is being produced. So when you use dantrolene, what it does is that it will basically stop this rhodine receptor, which is hyperactive, and so the calcium will not be able to move out of the rhodine receptors. Okay, and so it relieves muscle rigidity, it will correct the hypermetabolism, it will be able to reduce death. Okay, in malignant hyperthermia, it will be able to reduce the tachycardia and hyperthermia, and basically all these effects. Does that make sense? Where are you not okay? So rhodine receptor is basically allowing calcium to leave the sarcoplasmic reticulum. So it's without the rhodine receptor, calcium cannot leave the sarcoplasmic reticulum to cause all these effects that we've talked about. So in malignant hyperthermia, there's a problem. There's a mutation in the uh, rhodine receptor gene, the gene that forms the rhodine receptor. And then it is a gain of function mutation. Like the rhodine receptor is working too much. It is allowing too much calcium to leave the sarcoplasmic reticulum, and that's what is causing the problem. So if you give the drug dandrolene, which is we've been told, it will inhibit this guy from allowing calcium to leave the sarcoplasmic reticulum, And since calcium is no longer leaving the sarcoplasmic reticulum, and that is what was causing the problem, it means that you have excluded the problem. So the problem is no longer there. I don't know if that makes sense. Okay. Number 9 is saying state any two functions of iron in the body. Describe the mechanism of intestinal iron absorption. Well, iron has got many functions in the body. Iron can actually be converted from iron 2 plus to iron 3 plus, and that is what facilitates redox reactions that can be used for many things in the body. So iron can transport oxygen because iron is actually a component of hemoglobin. Iron can store oxygen because iron is a component of myoglobin, which is found in muscle. Okay. Iron can also be used in energy production because it's a cofactor in the mitochondria for many enzymes like the cytochromes. Remember in biochemistry you learned the oxidative phosphorylation and the electron transport chain. The cytochromes contains these are enzymes, and enzymes for them to function they need cofactors, and iron acts as a cofactor. Okay, and then iron is also needed for DNA synthesis. Okay, because the enzyme ribonucleotide reductase is this guy actually requires iron and is involved in DNA synthesis. Okay, and that's what actually causes a problem because if you don't have iron, it can also cause anemia because you can't form the red blood cells. And then it's iron is also important in immune function because it helps in proliferation and maturation of the immune cells. And then it also has got enzymatic reactions. So iron is involved in collagen synthesis, hormone production, and such. So there are main functions of iron. But then how does it get absorbed? In the intestines, the mechanism of absorption. So in the intestines, you look at it like this. This is the lumen of the intestines. When you eat your food which contains iron is going to be here. And then I'll just enlarge one cell this side. Okay, this is the lumen where the food is. And then this is just the epithelial cell which is involved in absorption. Okay, this is the epithelial cell. And then of course you are going to have a blood vessel this side. So anything that you eat has to first go into the epithelial cell and then it can now go into blood to go and start working. Okay. So now what happens is this. When we eat, iron can either be found as heme iron. Okay. Heme iron comes from animal sources. And then you can have a non-heme ion which comes from plant. So these two are absorbed differently. The heme ion which comes from animal sources is absorbed via the heme carrier protein 1. Okay, so heme carrier protein 1. While the non-heme ion is normally found as iron 3 plus. So it has first to be changed. This one which comes from plants, it has to be changed to iron 2 plus. So you have got an enzyme in the duodenum because that's where absorption happens, known as duodenocytochrome B. Uh, so this is the one that will be helping in that function together with vitamin C. Okay, so so in other words, when you eat food, let's say someone has got anemia, and then you are giving them iron to treat that anemia. You give them together with vitamin C because vitamin C is important in the absorption of iron. So that's the rationale. As you do medicine, things begin to make more sense when you know the way the body works. Okay, that's why we give vitamin C together with iron, especially for ladies because of menstruation and such. You get to lose a lot of iron. Okay, so that is what happens. Please take note of that. So once the iron has been absorbed, remember this side we have got the heme carrier protein 1. Okay, that will be absorbing the iron. So once the iron has been taken by the enterocyte, this same cell which I've called the epithelial cell is the enterocyte. Okay, the iron 2 plus is now going to enter via what we call a divalent metal transporter 1. Divalent metal transporter 1. Okay, let me see if that is showing. Okay, so you can see for this picture that is here, you have got this guy here which is a divalent metal transporter 1 that will be able to allow the iron to move from the intestinal lumen into the cell. But of course, we also mentioned of this guy here. You can see there's another guy there. Okay. So you have got the divalent metal protein there, and then you also have the heme, the HCP1. So both of them are there. They all both allow the ion to enter into the cell. Okay. All right. So here I showed the HCP1, which is heme, the heme. Uh, well, why is this name going? The heme carrier protein 1. And then you also have a divalent metal transporter 1, DMT1, that also allows calcium. So just the same here, the DMT1, okay? Divalent metal transporter 1. Why are we calling it divalent? Because iron has got a valence of 2. It's ion 2 plus. Okay, so that is what happens. And then once that ion has entered the epithelial cell, in this case the enterocyte, is going to be broken down. Okay, it's going to be broken down, releasing ion 2 plus. So in this case, we are considering that the ion that has entered is the ion 3 plus. So the ion, the actually the heme ion is what we are talking about in this case first. Heme ion, this guy that is here that you get from animal sources, once you get to absorb it, it's now going to be broken down by this enzyme heme oxygenase, and then you release the ion. Okay, and then that ion is now going to be taken. Besides that, the ion that has now entered the cell here, some of the ion is going to be stored, so it's converted into what we call ferritin. That's the storage form. So the ion is stored, okay, as ferritin inside the enterocytes. And then some of the ion is going to be transported to the other side, the basolateral end. And this side, there is a transporter which we call ferroportin. That's a guy that is going to allow the ion to be able to go that side. Okay, it's an ion exporter. The ion 2 plus is going to be converted back into ion 3 plus by hephaestin or ceruloplasmin. Once it is in the form ion 3 plus, it can now bind to what we call transferrin. Transferrin is a guy that transports ion. Okay, and then it to be able to go to the rest of the body. Now, what happens that the liver releases an enzyme which we call epsidin. What happens that when you have got too much high levels of iron in the body, this epsidin is going to come and bind on the ferroportin to prevent the iron that is in the food from being absorbed. And that is how regulation happens. Please take note of that. Okay, take note of that. So you can see what we have just been talking about. This iron that is here can be stored as ferritin. Okay, it can go to be used in the mitochondria as a cofactor and many other functions, or it can pass through this ferroportin. You can see still iron 2 plus, and then to be now converted to iron 3 plus, and then bound to transferred by transferrin. When you have got a lot of iron in the body, the liver will produce epsidin, which will come and bind here. Okay, and that's what you can see, it has bound there. So it means that the ion can't be transported to the other side because ferroportin is being inhibited. So that is what happens. That's how iron is absorbed in the body. Does that make sense?
Yes, it does. Now on that is hepcidin. You said it binds to what?
To ferroportin. Ferroportin, this protein that is present on the basal end of the enterocytes.
Okay.
Number 10 is saying body fluid compartments enumerate forces concerned with dynamic or fluid exchange at capillary level. Okay, this is very direct and simple. Okay, so when you have got a capillary, So when we have got a capillary is where gaseous exchange happens. Not gaseous exchange, but that is where basically exchange of nutrients and the things will happen. So this is the inside of the capillary. So you have got blood there, and then this is called the interstitium or the interstitial fluid. So you have got some forces that will be pulling. So as when blood is moving, there is the end of the capillary that is receiving blood. We call that as the arterial end. The end of the capillary that is taking away the blood, that is known as the venous end. So there are forces that keep things within the blood vessels and forces that pull things away from the blood vessels. So in the blood vessels, you have got proteins that keep things inside. We call the force that they generate to keep fluid inside, we call that as the oncotic pressure. Because that pressure is due to plasma proteins, we call it the plasma oncotic pressure. It's also known as osmotic pressure. That's a symbol. Okay, plasma osmotic pressure. And then there's a pressure that pulls the things away from the vessels. All right, we call that as the hydrostatic pressure of the plasma. So that is known as plasma hydrostatic pressure. So hydrostatic pressure is pushing away, oncotic pressure is keeping. And I've said oncotic pressure is because of proteins. The specific protein here is albumin. So if someone has got a liver disease, albumin is not produced. So what does that mean? It means that you can't keep water within the blood vessels. So the water will start leaving the blood vessels. So someone is going to have edema. Okay, they'll be swelling. So that is why you are going to see someone with edema if they have got liver disease. Kidney disease can also have edema, heart disease can also have edema. But anyway, we'll explain that for another topic. On the venous end, also you have got forces that are keeping. Uh, so I've talked about two things. I've talked about forces to do with the blood itself. These are the two that I've written. But then you also have forces to do with the interstitium. Okay, these forces that I've written here is to do with the blood. How about forces to do with the interstitium? Okay, so there is actually a force in the interstitium that wants to keep fluids within the interstitium. We call that as the interstitial oncotic pressure. Okay, interstitial oncotic pressure. And then there's also a force that wants to push things away from the interstitium. We call that as the interstitial hydrostatic pressure. Okay, so those are the two. So you can see that for the oncotic pressure or the interstitium, it promotes things from leaving the blood vessel. Look at where the arrow is going. Oncotic pressure of the interstitium promotes things from the leaving the blood vessel. While the oncotic pressure of the blood or plasma promotes things from being within the blood vessel. So therefore, we've talked about that. So it's saying enumerate the forces concerned with dynamic or fluid exchange at capillary level. So these are the forces. And you can calculate what we call the net filtration pressure. So net filtration pressure, the way we calculate it is you get all the forces that are pulling things away from the blood vessels. So that is the hydrostatic pressure of plasma. I can start with the H and then write the P. And then you are going to add another force that is pushing things away from plasma. That is the oncotic pressure of the interstitium. Okay, so we'll get these two values, we add them, and then we'll subtract those pressures that are keeping fluids within the blood vessel. So I've got the oncotic pressure of plasma, and then plus the other pressure is the one keeping things within the blood vessels is this one. It's the hydrostatic pressure of the interstitium. Now, normally, because oncotic pressure is because of proteins, normally in the interstitium, we don't have proteins there. So you discover that this interstitial oncotic pressure is normally a zero because we don't have proteins there really. Okay, that is how we calculate these things. Does that make sense?
Yes, it does.
Okay, good. 11 is saying define a reflex using the diffusion reflex as an example. Identify the components of the reflex arc. Now, a reflex is an automatic. Okay, it's automatic, it happens by itself and it's involuntary. Okay, specific response to stimuli, and it is mediated by what we call a reflex arc. So that's a reflex. Okay, that's a reflex, an involuntary, automatic, stereotyped. It can be beyond normal, and that's regulated by reflex arc. So for a reflex arc, there are things that you need to know about it. So the spinal cord has got the front and the back. So this is the back, this is the front. Okay. So the back of the spinal cord is going to be, okay, let's start with the front. So there's something like this which we can draw. So the back is responsible for sensations. It's sensory. Okay, something like that. So sensory inform you have got basic components of a reflex arc. There is the afferent, and then there is the efferent, and then there is the center. Okay, those are the basic components. My pen is just refusing from writing. Okay. So what happens is that you are going to have nerves that come from the back. These are sensory in nature. They are like that. And then you have got those nerves that come from the front. These are motor in nature. So this is sensory. Sensory means that it goes away from the spinal cord to an organ, and then the motor takes information from an organ. Actually, this is opposite, isn't it? Sensations go away from an organ. That's why we call those as afferent nerves. Because you are sensing, let's say for example, you touch something that is painful. You should be able to feel the pain. So that's a sensation. It comes to the spinal cord. Okay. So this is an afferent. Afferent is towards, so towards the central nervous system. And then the spinal cord itself, that's where you're going to have, we refer to that as the integration center. Yeah. This is where the information is going to be integrated, integration center. And then you are going to have the efferent, which will go back to that organ. So efferent nerves, these are motor. Okay. This is sensory. But besides this, what will be being carried by the sensory nerve, there's a stimulus. There's actually something that caused the stimulation of the sensory or the afferent nerve. So there's a stimulus or stimuli. And then the efferent nerve is also going to go and innervate the muscle of interest. So we call this as the effector organ. So these are the components of the reflex arc: stimulus, sensory, nerve, efferent. There's also the center here, and then the efferent, and then the effector. So specifically for the diffusion reflex, the stimulus here, what stimulates diffusion is the distension, okay, like the rectum. If it distends, what causes distension is because of the fecal matter that has accumulated. So you have got stretch receptors there. So it's the stretch receptors, which are mechanoreceptors, where the stimulus will bind. Okay. In this case, it is the stretch sensation. Okay, so that is what happens. So because of stretch, that is going to stimulate these stress receptors, and then information is going to be carried by the afferent or the sensory nerves via what we call the pelvic nerves. And this pelvic nerves is from S2 to S4. Okay, the pelvic nerves. So this information is going to the spinal cord. So it has started. Here is the rectum. It has been stretched. So this nerve is going to go to the spinal cord, to the back of the spinal cord. And then from there is an integration center just within the spinal cord. Okay. That's known as a spinal diffusion center. And then from there the information has been processed, and then motor information is going to be sent back. And this information is actually parasympathetic in nature, because you actually go to the toilet because of the stimulation of the parasympathetic. Sympathetic is stimulated when you're either running, there's a fight or flight. When you're running, you don't want to go to the toilet, you don't have that time, you're concentrating on running. So the parasympathetic is what is activated when you want to defecate. And there you have got also the pelvic nerves, okay, that are going to now go to the colon. So this one was picking up sensation, and then you are now having those other ones that are going to the colon, all right, and also the rectum. At the same time as that is happening, you also have the somatic nerves. Somatic means that they control skeletal muscles, because remember, reflex act basically will control is involuntary. Okay, but there's a part where there are skeletal muscles. Skeletal muscles are normally voluntary, but because it's an arc, it's involuntary itself. So there's a nerve which is called the pudendal nerve. Okay, and that is also S2 to S4, because it's coming from the same spinal segment. This one was sensory in nature. It was just a sacrospinal nerve. This one, which is motor from S2 to S4, specifically is known as the pudendal nerve. It's going to go and innervate the external anal sphincters, and then someone can be able to defecate. So what happens that the effector organ in this case is going to be the smooth muscles of the rectum, even the internal anal sphincter. Those ones are going to, once this nerve goes back, it will go back to the smooth muscles of the rectum and the internal anal sphincter. The internal anal sphincter is involuntary, you don't control it. So the rectal is going to contract to push the feces away, and then the internal anal sphincter is going to relax so that it can allow the contents, okay, the feces to move. Now the activation of the pudendal nerve, this one is motor in nature, is going to actually go to the skeletal muscles, specifically the external anal sphincter. The external one is voluntary, you control it yourself. So it's not because of a reflex. That is why for a defecation reflex, even if you feel the edge of going to the toilet, you're not you can control yourself, because you've got the external anal sphincter that you can control. Meaning that if there's actually a problem with the external anal sphincter, or a problem with the nerve that innervates this guy, someone can't control themselves. For example, children, this guy is actually weak, so they can't control themselves. Okay, so these are the components of the reflex arc. Does that make sense?
Yes, it does.
Okay, and then the last question for now: what is the effect of the following two agents on neuromuscular transmission? Neostigmine and the botulinus toxin. Okay, this has to do with the parasympathetic nervous system. So the nervous system is divided into somatic and the autonomic. Somatic is basically the one you control. Autonomic you don't control. Autonomic is divided into sympathetic and parasympathetic. The main hormone in the sympathetic nervous system is known as epinephrine and norepinephrine. They're also known as adrenaline and noradrenaline. Parasympathetic nervous system, the main hormone released here is acetylcholine. Okay, acetylcholine. Now, when this acetylcholine has been released, what happens is this: you have got, this is a nerve. Acetylcholine is hidden in vesicles. So what happens is that when an action potential reaches, because it's coming from the body, okay, can you just turn off the mic? Okay. And then you can switch it on anytime you want to ask anything or contribute. So this side you have got the body of the neuron. That's where action potential starts. When the action potential reaches, this is the terminal of the neuron. It does is that it will cause calcium to enter the cell. And then this calcium is actually going to come and promote this vesicle that is containing acetylcholine to come and fuse with the membrane here, and then the acetylcholine is going to be released. The acetylcholine now will be able to bind on the receptors. It can be muscles. Okay, it can be a muscle. It will come and bind here. And then contraction is going to happen because once acetylcholine binds on the muscle here, action potential is going to be generated. And then the procedure is going to go as we described in muscle contraction for the questions on top. Okay, that is what happens. But then there are some drugs that can inhibit here. Okay, so there are some drugs that we can give that can work like acetylcholine. So we call those as cholinergics or acetylcholine receptor agonists because they work on the receptors like acetylcholine. Now you have got two types because after acetylcholine has actually bound on the receptor here, what happens is that the acetylcholine is supposed to break down. It breaks down and that's how it stops working. So in other words, there is an enzyme here that causes the breakdown. So if and we call it the acetylcholine estrase. Acetylcholine estrase. So if you can just inhibit that enzyme that breaks down acetylcholine, it means that it will still have more function or more time to work on the receptor. So it will cause contraction to continue happening. So there are some drugs that we call acetylcholine estrase inhibitors. These drugs will basically stop the breakdown of acetylcholine so that it can continue working. Okay. So we have got two classes of drugs that work like acetylcholine. We call them cholinomimetics. They mimic acetylcholine. Mimetic is to mimic, to behave like. So you have got those drugs that can so you have got two classes of drugs. You have got those drugs that can basically come and attach on this receptor and then work the way acetylcholine is supposed to work. And we call those as direct acting cholinomimetics. We also call them, because acetylcholine is in the parasympathetic nervous system, we also call them direct acting parasympathomimetics. And then you have got these drugs that will basically come and stop the breakdown of acetylcholine. Acetylcholine will still not be broken down, so it will still be working. We call these ones as indirectly acting parasympathomimetics. So what do? They just inhibit acetylcholine estrase enzyme so that it doesn't break down acetylcholine. The direct one, no, those were binding on the cholinergic receptor itself, behaving as agonists, behaving like acetylcholine. So examples of the direct acting include acetylcholine itself, carbaco, bethamico, you have got methacholine, masculine, pilocarpine, nicotine. Okay, so that is what happens. These ones will promote contraction. The indirect acting includes physostigmine, neostigmine, pyridostigmine, androphonium. So these ones will basically stop the breakdown of the enzyme. I mean the breakdown of acetylcholine by acetylcholine estrase so that it can work more. Okay. So these drugs are going to be used in those conditions where there is weakness, like someone feels so weak, contraction is not happening because acetylcholine promotes contraction. But now there are some other drugs. For example, there's this drug which we call botulinum. It happens oppositely. What it does is that it prevents the fusion of acetylcholine vesicle with the cell membrane. So it means that acetylcholine is not going to be released. That's what botulinum does. And this guy is actually a toxin. It is produced by a bacteria which is known as Clostridia difficile if you learned in bacteriology. Okay. So that bacteria, that's what it causes. So because acetylcholine is not being released and it causes contraction, so it means that there's going to be relaxation. So the botulinum toxin causes what we call uh, flaccid paralysis. Someone is going to be weak. They are floppy, like as though they don't have muscles. They are so weak. So that is what happens. So botulinum toxin, this guy is going to block the release of acetylcholine from the presynaptic nerve. This nerve is before a synapse. We call it presynaptic cell. And then this cell where the hormone will bind, we call it a postsynaptic because after a synapse or after meeting. That is how that guy gets to work. So it causes flaccid paralysis. Okay, when do you use this guy? Basically, conditions where someone is weak. Okay, I mean a condition where someone is overly contracted so that they can be relaxation. Neostigmine is the opposite. It's used in those conditions where there is weakness so that contraction can happen because it's the opposite. Remember, neostigmine was promoting inhibiting acetylcholine stress so that acetylcholine can still be able to work. It can still cause contraction. So these drugs are going to be used in conditions such as myasthenia gravis. Okay. So that is what happens. Of course, you have got other drugs just here because for you to form acetylcholine, you need acetyl-CoA and choline. But choline comes from the outside of the cell, and there is a transporter here that is required. So there is a drug such as hemicolinium that stops the uptake of choline. So this will still lead to acetylcholine not being produced, and so it will not be able to work. There's also what we call the vesamico. It's another drug. Vesamico basically stops acetylcholine from entering into a vesicle, and that's how it works. So because there's no acetylcholine in a vesicle, even if the vesicle is merging with the cell membrane, well, there's no acetylcholine that is going to be released. So that is what these drugs do. Okay, basically that.